Imagine this. You've just finished synthesizing your new drug molecule. The chemistry looks perfect, the purity is high, and the yield is excellent. But before that compound can ever reach a patient, there's one invisible factor that could stop everything in its tracks: residual solvents. These are the hidden traces of organic chemicals left behind after synthesis, crystallization, or purification. They may not affect how the drug works, but they can affect how safe it is. So today, let's explore pharmaceutical residual solvent testing. What it is, why it matters, and how drug developers can ensure their products meet global safety and regulatory standards. Residual solvents are like silent passengers in your drug product. They come from the manufacturing process. Solvents used to dissolve reactants, purify intermediates, or recrystallize the final API. And while most of the solvent evaporates during processing, tiny amounts can remain trapped in the crystal lattice or formulation. Here's the problem: even trace amounts can be toxic. Some solvents cause liver or kidney damage, others are carcinogenic, and some interfere with drug stability. That's why regulatory agencies, the FDA, EMA, and ICH, require every manufacturer to test and control residual solvents in accordance with ICH Q3C guidelines. Now, ICH Q3C, or the International Council for Harmonization Guideline on Residual Solvents, provides the foundation for global solvent control. It classifies solvents into three main categories based on their potential risk to human health. Let's break them down. Class 1 solvents. These are the red list solvents: carcinogenic, mutagenic, or environmentally hazardous. Examples: benzene, carbon tetrachloride, 1,2-dichloroethane. They must be completely avoided in drug manufacturing. Class 2 solvents. These are more common but have potential toxicity if present above certain limits. Think methanol, toluene, or acetonitrile. The FDA sets PDE limits, permitted daily exposure, for each. Class 3 solvents. These are considered low risk with higher allowable limits. Ethanol, acetone, ethyl acetate. Solvents like these are acceptable up to 5,000 parts per million. So, in every drug submission, NDA, ANDA, or DMF, developers must prove that their product meets these ICH Q3C limits. Detecting residual solvents isn't just about running a simple test. It's about precision, selectivity, and sensitivity. The gold standard technique is gas chromatography. Why GC? Because it can separate volatile organic compounds based on their boiling points and interactions with the column. It's fast, accurate, and capable of detecting solvents in parts per million levels. There are several common approaches. Direct injection GC, ideal for APIs that dissolve well in a compatible solvent. Simple, but not always suitable for thermally unstable materials. Headspace GC, the most widely accepted method. The sample is heated in a sealed vial, and volatile solvents are analysed from the vapour phase, leaving non-volatile residues behind. GC-MS, used for unknown or unexpected solvents, providing structural identification and confirmation. Each technique has its own advantages, but Headspace GC remains the regulatory benchmark for residual solvent testing. Now, let's talk about the real-world challenge: developing and validating a reliable residual solvent method. It's not just about meeting limits, it's about building confidence in your results. Here's how expert labs like Resolve Mass Laboratories approach it. Step 1. Solvent selection. Identify potential solvents used at every step of synthesis: reaction, purification, and formulation. This ensures no residuals are missed. Step 2. Sample preparation. Select an appropriate diluent that won't interfere with GC analysis. Typically, DMSO, DMF, or water. Step 3. Method optimization. Adjust oven temperatures, column types, and carrier gas flow to achieve clear separation between peaks. Step 4. Validation per ICH Q3C. Evaluate specificity, accuracy, precision, linearity, limit of detection, and quantitation. Each parameter ensures regulatory compliance. When done right, these methods can detect solvents at levels far below regulatory thresholds, often under 10 parts per million. But even with the best equipment, there are challenges. Sometimes solvents co-elute, meaning their peaks overlap, making quantification tricky. Other times, residual solvents bind tightly within the crystalline API and require special heating or derivatization steps to release. And occasionally, a change in synthetic route introduces a new solvent altogether, requiring method revalidation. That's why working with an experienced CRO or analytical lab can make a major difference. Their expertise ensures not just compliance, but efficiency, saving developers from costly rework during FDA review. From a regulatory standpoint, it's not just the test results that matter, it's how you document them. Your residual solvent testing must include a comprehensive solvent list from your manufacturing process, a validated analytical method, often headspace GC, chromatograms and raw data supporting identification and quantification, summary tables showing results versus ICH limits. For ANDA and DMF submissions, the FDA expects this data to demonstrate that your process consistently meets Class 1, 2, and 3 limits, even across multiple batches. Residual solvent testing doesn't have to break your budget, but it does require smart planning. Developers often reduce costs by testing early, during R&D scale-up, rather than waiting for validation or pre-approval inspection. Working with a qualified analytical partner also ensures consistent data traceability and FDA audit readiness. Some labs, like Resolve Mass Laboratories, offer cost-effective, GMP-compliant testing with full regulatory documentation, including chromatographic data, method validation reports, and certificates of analysis ready for submission. At the end of the day, residual solvent testing is more than a regulatory checkbox. It's about patient safety and scientific integrity. Every peak on a GC chromatogram tells a story about how carefully a drug was made, purified, and controlled. For developers, mastering solvent testing means mastering one of the most fundamental aspects of pharmaceutical quality assurance. So whether you're developing a small molecule API or a complex peptide drug, make sure your residual solvent data tells the right story, one of precision, compliance, and trust. For guidance or analytical support, reach out to a qualified lab partner like Resolve Mass Laboratories, where science meets compliance.